Before starting any of your processes, you need to ensure that you've undertaken hand decontamination through hand washing, and that you're wearing all the required personal protective equipment that is appropriate for the task you're about to undertake. These will be listed in your standard operating procedures and will include items like lab coats, gloves, goggles, and masks as needed. Once you've finished with the hand wash and put on your PPE, you'll need to prepare your work areas. This includes taking out any of the consumables that will need to be brought to room temperature, as well as cleaning down your workspace. You'll also need to do things like prepare disc pots that contain 10% bleach. Your workspace must be cleaned at the start and end of every day, as well as in between changes of procedure when you'll be working with different specimen types. The same process will also need to be undertaken if there are any spillages. For fluid spillages, you'll need to follow local guidelines, as there may be additional steps that you're required to undertake. Preparing and cleaning down of your workbench includes not just cleaning down the bench space itself, but any of the equipment you're likely to use, including racks and pipettes. We are now going to look at the preparation of the LAKANA collection bags. Firstly, we will put together the nasopharyngeal collection bags. Then we will put together the rectal sample packs. If you are pre-aliquoting your prepared media with 1 ml of media into 2 ml screw top vials, take them out of the fridge in order for them to be at room temperature. Check for your STGG media that it is labelled on the top, on the side, and is within expiration date. Also check that the media is clear and does not appear to be contaminated. Then do the same for your CB Carrie Blair media, i.e. labelled on the top, side, is within expiration date, and is clear and does not appear to be contaminated. Finally, do the same for your DESS media, i.e. labelled on the top, side, is within expiration date, and is clear and does not appear to be contaminated. You will also need some dry 2 ml screw cap vials in order to complete your collection bags. The first pack we are going to assemble is a nasopharyngeal pack. Place a blank sticker on a plastic sample bag and label it with the letters NP for nasopharyngeal. You will need to add a linked barcode for the collection packs to all of your vials that you're going to include. If you have pre-labelled and pre-aliquoted your collection vials, you can add your checked STGG tube and a nasopharyngeal copan swab 553C or 503CS01 into the NP plastic bag and close. Then you will need to assemble your rectal pack. Take a zip bag with a blank sticker, which you then label rectal. Then affix a pre-printed unique barcode to your CB Carrie Blair tube. A matching group-linked unique barcode label to your DESS tube. And add another to your empty 2 ml dry vial. The barcodes which we're attaching in this video may not be the same as the ones that you're using as part of the study, so please bear that in mind. Once you have put your checked, labelled and barcoded CB, DESS and dry swabs into the rectal sample bag, add 3 swabs with 30mm breakpoints. These are the Dewey swabs or Copan swabs, 520CS01, and then seal. We will then assemble the total sample collection bag. Place a blank sticker on the large Ziploc bag and label collection bag. Place your labelled NP and rectal collection bags into the large collection bag, making sure that all vials are labelled and within expiration date as previously stated. The collection bag is then ready to be stored at 2 to 8 degrees in the fridge and can be sent out to CS Coms and from there to the villages. You will then need to clean down your bench space appropriately.